Hello, my name is Mike and I'm going to lead you through a tour of our lab. I'm an associate professor in the biochemistry department and the Biotechnology Institute at the University of Minnesota. And we have a MAISRC funded project that focuses on developing a new class of technology to control invasive species called genetic biocontrol. Now, if you've heard or read in the news lately about engineered mosquitoes being released out in Florida, then you've heard of genetic biocontrol. It's a new technology, a very powerful new technology, in which the pest organism is genetically engineered to become the pesticide. In essence, we engineer male organisms, in our case, common carp, that we, in the future, would release to the wild. Those engineered males would mate with the wild females and spread deleterious genes through the population only of common carp. Because it relies on mating to spread these genes, there's a very low chance that it would negatively impact native fish or other fauna, flora and fauna. And by spreading those deleterious genes, we can suppress or even eradicate a local population of invasive species. So we think a lot about DNA in our lab. We have to engineer these somewhat complicated genetic blueprints and insert them into the genome of carp. And I'm going to walk you through the lab today, and some students will help me out and show you the places in the lab that we do the genetic engineering, where we rear the carp, and how we insert DNA into their genomes. It all starts using computers. We look at the genome sequences of carp, we look at the genome sequences of other organisms that we can take genetic elements from and combine them into an engineered genetic blueprint that we then will order from a DNA synthesis company. There are DNA synthesis companies all over the country, and a few days after we order it, the DNA arrives in a test tube like this. So this is man-made DNA that's fabricated precisely to the blueprints that we specified on the computer. The next step is introducing this synthetic DNA into the genome of carp. And for that, we need fresh carp embryos. So next, I'm going to hand the baton to Nick Jacob, a graduate student in my lab, who's going to walk you through our space at the MAISRC Containment Lab and show you how we rear adult carp and induce them to spawn all throughout the year to generate these fresh embryos for our genetic engineering experiments.
Hey everyone, Nick Jacob here. We are now down in our MCL space. We've had some exciting developments over the past couple of years, including the ability to reproduce carp off-cycle, meaning we can get carp to spawn at any time of the year that we want. Now, to give you a quick primer on carp reproductive biology, carp usually spawn from late May into early June, and then spend the entire summer redeveloping their gametes for the following spawn. Now, when fall comes around, carp get environmental cues such as decreasing photoperiod and decreasing temperature to put their gametes into what's called an arrested state. And these gametes stay in this arrested state all the way through the winter until spring, when photoperiod and temperature increase again and they get the cue to spawn. Now, what we've been able to do in lab is artificially change the photoperiod and water temperature to trick carp into spawning at different times of the year. And through this, we can get year-round production of eggs and sperm for our transgenesis experiments. Now that I've shown you kind of how we do what we do, I'm going to take you on a tour through the system and show you the different components. So our MCL space has four main components. The first is a summer and spring rearing system, which are these tanks you see right here in the middle. The second is an overwintering system behind this black curtain. The third is our larval rearing system, which we use after our transgenesis experiments along the scaffold. And the fourth are our juvenile tanks. Now, we have fish at different parts of our MCL system in the spawning process, and so I want to show that to you right now. In this tank right here, we have some fish that we just spawned in the spring, and right now they're being kept at warm temperatures so that they can redevelop eggs and sperm. On the next tank right over here, these are fish that we just brought up from our wintering process, and you can see they're a little bit more sluggish. They should be ready to spawn in about a month. Now I'm going to take you outside and around to our overwintering system, which is kept over here. We keep our fish behind this black curtain so that we can decrease the photoperiod and the temperature without other fish being affected. These fish have been at cold temperature for about a month, and in about a couple months, they'll be ready to be brought up to warmer temperature and spawn. Now, if we wrap back around to our main system, I'm going to introduce the juvenile tanks. Right now, we have 4 juvenile tanks, but we're getting many more soon. Each tank is composed of a different genotype of fish, meaning that each tank has fish that contain different genetic information. Some tanks, such as these tanks, contain GFP fish, meaning they contain a gene that makes them glow green. Some of these fish are just pure wild-type fish from different genetic backgrounds. So now that you've seen our MCL system, I'm going to kick it over to Julie Benkin, another graduate student in our Szymanski lab, who's going to show you what we do with the eggs and sperm once we harvest them from our carp.
Hi, I'm Julie Benkin from Mike Szymanski's lab. Once we have the eggs and sperm from the adult carp, as Nick was just describing, we take them into this lab, where we have equipment to insert the synthetic DNA into embryos. The first step is to take the eggs and the sperm and combine them in water in order to create embryos. These embryos would naturally develop into wild-type carp. However, we're going to, shortly after combining them, put them into a small dish like this, and then use the microscope and micromanipulator, this set up to the right here, and introduce the synthetic DNA to these carp embryos. The first step is to take a very fine glass rod and pull it into a glass needle with a very, very, very fine tip, such as this one here. After which we would load the synthetic DNA into the back of this needle, as well as any other components that are needed to make the gene edits. This glass needle with the DNA in it would then be put into the micromanipulator, which connects to a hose leading to this system. This system back here uses gas pressure to force the synthetic DNA out of the needle and into the embryo. At that point, it's a matter of using the micromanipulator in order to line up the needle with the embryo, looking through the microscope, and then very carefully insert that needle into the embryo. Once it's inserted, the person operating this steps on a pedal, which forces the gas pressure out of the needle and forces the synthetic DNA into the carp embryo. Once the carp embryo has that DNA in it, one just carefully pulls the glass needle back out and moves on to the next embryo. And once a plate of those embryos have all been injected, they're covered with water where they're allowed to incubate for a few days, after which they are assessed. Their survivorship is calculated, and they move on back into the MAISRC lab, which Nick was just showing you. So we're going to go back to Nick now.
Now we're back in our MCL space at our larval rearing system. Now after microinjection, our cohorts of fish are placed into individual tanks so that we can distinguish between the different genotypes that we create up in the lab. Now our fish are fed brine shrimp for 2 weeks until they can be weaned onto a commercially available carp diet. Now these fish will remain on this system for about 2 months until they can get large enough where we can insert what's called a pit tag into their abdomen. Now the pit tag contains a little code that we can scan with a scanner so that we can identify each fish that we create up at our lab. Now once these fish contain the pit tag, they can be moved onto our juvenile system and then entered into our main MCL spawning system. Well, that about wraps up our tour of our MCL space. Thanks for joining us virtually. We hope that you can come visit us in person soon.